thank you for the invitation to come to this amazing place, to this amazing conference. And congratulations to your organization with their 20th birthday. These organizations are very, very important for the development of psychiatry and psychiatric healthcare in a country. And I know that Turkey, for example, is doing a very good job where ADHD is involved. So with that being said, I indeed want to talk about neurobiology of ADHD across the lifespan. I saw the motto of your conference, Exploring Convergence of Psychiatry, Neuroscience, and AI, and I hope I can deliver on that. I'm a basic scientist based in the Netherlands, and I'm going to take you through some of the research that we've been doing in the context of many international consortia also on ADHD neurobiology. So as I said, I'm really, molecular biology is my thing, is my what I have been trained in, but still I have the ambition to improve healthcare for people with psychiatric conditions and especially ADHD. And I think I can contribute to that by helping understand what ADHD is in the neurobiological sense. And also, over the years, I have become a little bit more ambitious in my wishes or my goals, and I want to contribute to a more biologically based nosology in psychiatry, and I think it is also very, very important to bring together many different disciplines when we're talking about research on ADHD. It's not enough to just think of good research projects as researchers, but we have to work closely together with people with lived experience and many, many of the other stakeholders as well, from the get-go, from the design phase of studies. So today we're talking about ADHD, and ADHD is a very, very controversial condition diagnosis still in the field of psychiatry, and many say ADHD is a product of our modern times. But then if you look at the history of ADHD, you can see that the first descriptions of symptom clusters that look like ADHD come from already 1775, for example, in a German textbook and shortly after in a Scottish textbook. So ADHD is not a new diagnosis. It's been around for a very long time. Still, what is it that makes it so controversial? Well, on the one hand, I would like to say that patients and their representatives have fought very, very, very hard to get ADHD established as a diagnosis and to get help for ADHD. But then we have a lot of people having ADHD here, 5% in children, 2.8% in adults worldwide, and still the public is skeptical of the fact that ADHD might exist. And more importantly, they are also uninformed of what ADHD is. You hear still a lot of times ADHD is a childhood condition. It goes away when people become adults. Diagnosis is often associated with stigma, and it also is a problem for people to participate in society fully. So you have to do extra tests for your driver's license. You have restrictions when choosing an occupation. So it's not necessarily popular to have ADHD. That being said, we know that ADHD medication is one of the most efficacious medication in the whole of medicine. It does dampen only the symptoms. It doesn't cure ADHD, but patients still are not keen on taking ADHD medication for longer periods of time. We also know that several comorbidities of ADHD and other problems that occur in ADHD, like suicidality and also criminal behavior, are preventable if we diagnose early and if we treat. And this is, for example, nicely illustrated in a paper from the group of Henrik Larsson from 2019, where they looked at the short-term treatment effects of ADHD medication, where you can see that It treats also injuries and traumas, motor vehicle accidents, criminality, suicidality, substance use disorder, depression, etc., etc. Even seizures can be treated, can be prevented if ADHD is treated. At the same time, people with ADHD in many countries don't necessarily feel themselves as patients. They want to be part of society. There's a huge neurodiversity movement in autism, but also coming to ADHD. On the other side, we have a lot of countries where ADHD is not an established diagnosis, at least not in adults. So we cannot treat people with ADHD in these countries. So there are huge differences between countries. So, well, I'm not telling you anything new on this slide. The only point that I want to make is that still, when we're talking about a diagnosis of ADHD, we're talking about a diagnosis being made based on clinical interviews and questionnaires. We do not have objective markers of ADHD that we could use to support the diagnosis. And this is the same as in other psychiatric disorders. Importantly, if you look at ADHD symptoms in the whole of the population, you see that it is sort of a continuum. Many people in the general population have a few ADHD symptoms. But only if you have a lot of these symptoms over a longer time, you will cross what we call here the diagnostic threshold, and you become a clinical case. You require a diagnosis and you require treatment. But still, what this means is that people here, the clinical cases, are not qualitatively different from people in the general population. They just show more ADHD symptoms. ADHD in itself, based on its symptoms, is a problem for people. But together with that comes a lot of additional comorbidity. And this is comorbidity with other psychiatric conditions, but is also comorbidity in terms of neurological diseases, even neurodegenerative diseases, cardiovascular problems and metabolic diseases, as well as immunological and inflammatory diseases are common in people with ADHD. And together with impact and ADHD across the lifespan, the ECMP network, we put together a few reviews on the lifespan perspective of comorbidity in people with ADHD. What about the brain in ADHD? A few years ago, Martine Hoogman and I looked at tried to get knowledge on the brain in ADHD, and we put together the magnetic resonance imaging studies on ADHD. What we found is that there is quite a lot of studies, but all of them are rather small, and many of them are not replicable. The findings are not, many of those are not replicable if you look in another cohort. So what we did is actually bring together across the field all the data that was out there on ADHD neuroimaging, and we did that in the context of the Enigma consortium, and we founded an Enigma ADHD working group, which is still led by Martina and me. Putting together data on over 2,300 people with ADHD and over 2,000 healthy individuals, we found a number of differences in brain structure in people with ADHD. And what we found was that several regions in the brain are slightly smaller in people with ADHD. And I've underlined here on average because the differences are small on the one hand, and they are not seen in everybody. And this is very, very important. And differences between people with and without ADHD are the largest in young children. So it really seems to be a developmental problem. And if people with ADHD become adults, they can still have ADHD, but we don't see the differences in their brain structure anymore. So what's happening there is still a question. And the regions that are smaller in children with ADHD also differ in people without ADHD depending on the number of ADHD symptoms. So again, here is this quantitative aspect of ADHD. Even if you don't have symptoms, the more if you don't have a diagnosis, the more symptoms you have, you have a slightly smaller brain region, number of brain regions. We asked the question based on this, is there a possibility to predict ADHD status based on the imaging data? And Yanli Zhang James from the group of Steve Ferrone took this forward into a machine learning study using the data of Enigma ADHD. And what she found is that, yes, we can predict the ADHD cases from neuroimaging data, but the accuracy of this prediction is rather low. We only reach about 65% accuracy, and this is too low to use in clinical settings. This is both for adults and for children very, very similar. So ADHD, importantly, is a disorder, a debilitating disorder in its own right, but what we also have to realize is that the problems that people with ADHD have do not stop at their symptoms. ADHD is often an entry point into a negative life trajectory where you find problems in educational settings, social, financial problems, lower socioeconomic status, multimorbidity, and an overall reduced quality of life. At the same time, we know that several of the ADHD-related problems can be prevented by an early diagnosis and adequate treatment. And even more, some people with ADHD thrive, and people with ADHD are overrepresented among artists, entrepreneurs, etc. So what is it, ADHD, and what can we do about it? Understanding the biological, social, and societal mechanisms underlying ADHD will really be crucial to prevent impairment in people with ADHD and harness potential strength that people with ADHD have. So how can we find out more about the biology? I'm arguing that genetics is really an excellent starting point into these biological studies because it's something that we are born with and it is not influenced, for example, by our experiences or by medication, for example. And ADHD is highly heritable. If you compare it with things like autism, schizophrenia, bipolar disorder, it is very similar in the percentage of ADHD phenotype that is explained by genetic factors. So then, how does that come about? This is because our genome, the genetic material that we carry, differs between individuals. We are largely the same, but we differ a little bit. And in this difference that we have, 15 million genetic variants differ with a frequency of more than 1% in the population, and over 30 million low-frequency variants are found in our genome. And these differences make us different. They determine whether we have black hair or blonde hair. They determine whether our eyes are blue or brown, etc. And they also determine whether or not we have an increased risk for ADHD. If we're thinking about heritability, we often think about single genetic variants that have very huge effects on disease risk. However, when we're talking about ADHD, what we're talking about are genetic variants that are very common in the population, so they are not restricted to people with ADHD. And they increase the risk for ADHD only by a very small amount. There are also additional low-frequency variants that we learn also have an effect on ADHD, but also those are relatively small in their effect on ADHD risk. So what we think ADHD looks like genetic is that those with ADHD have more genetic variants than those without ADHD. So essentially you have two different curves that only partially overlap each other. And what is shown here are 5 genetic variants that are more frequent in those with ADHD, but we know that we're talking more about tens or even hundreds of genetic variants that are different in people with ADHD and that have to come together to give you a very strongly increased risk for ADHD. And genetic factors do not work alone. They work in interplay with environmental risk factors. So what does that look like? What does this genetic risk for ADHD look like? Well, think about a biological process in the brain. Like, for example, here, the merging or the interconnection of neurons. This happens during early neurodevelopmental neurodevelopment. The neurons have to find each other for them to be able to communicate and to make our brain function as we know it. This outgrowth of neurons is orchestrated by a number of genes. And this is what I've shown here, a tower of genes that work together in this biological process. If the genes are, like here, quadratic, the tower is very stable. However, if there is a genetic variant, the tower is not so stable anymore. As long as you have only a few genetic variants like that, this is not a problem. But if you have many, then the tower really becomes unstable. And the process of neurite outgrowth of this neurodevelopmental process may be different. What could happen is that some brain cells actually form too short extensions and do not really communicate with the ones that they are supposed to communicate with. Or some of the brain cells happen to grow too slow and do not reach their target either. Or some brain cells do not grow directly towards their target. So these are things that could happen. Our brain is very well able to deal with such small differences. The brain is very, very flexible. Think about stroke, for example. If you have a stroke, many people recover many of their functions after a stroke. This shows the flexibility of our brain. However, if your cells communicate a little bit less well in the brain, this means that we are less able to adapt our behavior to the needs of an environmental situation that we encounter. And that could go along with ADHD symptoms. So trying to explain that in a different way. This is the burden somebody or the population carries for ADHD. A person can be here, for example, and then they have a very small burden for ADHD. And they can be here, and then they have a strong burden for ADHD. What does that mean for their behavior? If they have only a small burden, they have a large repertoire of behaviors. And here, for example, showing just the hyperactivity. They can be restful when it is needed. They can be active, very active when it is needed. Somebody who has a higher level of genetic burden, they don't have this full repertoire. They have a more restricted repertoire in their behavior, for example. And if you have a very strong burden for ADHD, a genetic burden for ADHD, then your repertoire may be even smaller. This is just heritability, just genetic factors. What about environment? There can be two scenarios. Your environment can empower you, can help you adapt to the daily situations, make you more flexible than what your genetic burden for ADHD may actually tell your body. On the other hand, the environment, if it is not supportive, can increase the burden further and can shorten your repertoire. So with the high heritability of ADHD, genetics gives us possibilities to understand biological processes that are involved in ADHD. And how do we do that? Well, we have to find the genetic factors that are involved. And how is that working? Currently, we're using genome-wide association studies for that. And this means that we're genotyping 200,000 to 1 million, over 1 million genetic variants in everybody in an individual to perform genome-wide association testing. And in these genome-wide association testing, we're asking the question whether genetic variants are more frequent in people with ADHD than they are in people without ADHD. We're doing this work in team science because, again, the genetic factors have small effect sizes. So we need huge sample sizes, like for neuroimaging. We need huge sample sizes to be able to have enough power for these studies. And for that reason, in 2007, the Psychiatric Genomics Consortium was founded to bring together all the data on psychiatric genetics to be able to do powerful studies. The most recent study that we did for ADHD was done in over 38,000 people with ADHD and over 186,000 people without ADHD. And what you see here is a Manhattan plot where on the x-axis you see the chromosomes, and every dot here is a genetic variant that was tested. On the y-axis you see the significance for the difference in the frequency of this genetic variant for people with ADHD and without ADHD. The higher these towers, the more significant is this finding. And what we found here were 27 towers and 27 genome-wide significant loci on the human genome that carry a risk factor for ADHD. And out of those, we could find or extract, based on epigenetics, for example, 76 credible ADHD genes. And with those 76 genes, we went further to find out how they influence the biology of the brain in ADHD. So how do we do that? We use this model as a basis where we have genetic factors up here, and these genetic factors together, several of those, influence biochemical processes. Biochemical processes influence cell function influences morphology, and all of that goes in a convergence model where we have many different genes involved, but we have one outcome, the disease symptoms. So how do we learn about these different levels? Looking at 76 risk genes, for example. Well, the thing that we're using most is bioinformatics. And in that, we are really fortunate that there is so much data now publicly available on the biology, on biological, that lends itself for biological analyses. For example, there is a whole collection called GTEx of gene expression data from many, many different tissues in the human body. And we can ask the question where these 76 genes for ADHD are predominantly expressed. And you may not be able to read it, but those findings that reach significance all tell us that ADHD genes are expressed in the brain. Well, I think this is what we would have expected, right? That ADHD is a disorder of the brain, and indeed, this is also what we find. Going further, we can also, based on single-cell gene expression data, we can also ask which type of brain cells are involved. And what we find here is that dopaminergic neurons, GABAergic neurons, and cells of the red nucleus are involved. Well, cells of the red nucleus, I had to look up, but the red nucleus apparently is a region of the brain that is very much involved in integration of motor data, motor information. So even using bioinformatics, we can come from genetic factors even down to the brain cells that are most likely the substrates of ADHD. We also can look at other types of biological processes that are involved, and this is work that we did quite early on, for example, with Geert Poelmans, where we put together all the data from different genetic studies and found that biological processes that were altered in ADHD were neurite outgrowth and synaptogenesis, so processes that take place very early in brain development, but also synaptic plasticity, and this is a process that we use every day to be able to learn, to be able to behave in our environments. And this actually is what we treat, for example, with methylphenidate. So both developmental processes but also acute functioning of brain are altered in people with ADHD. So when we look at our pyramid on its head, the convergence model, we see that the 76 risk genes lead us to neural outgrowth, synapse maturation, and function, lead us to dopaminergic and GABAergic neurons as well as glia. I told you about the imaging work that showed us that there is reduced subcortical volumes, reduced intracranial volume, and also reduced cortical surface in ADHD. What then can we do with such information on biological processes in ADHD and genetics? Well, first of all, I think it is important to communicate the information about the heritable component of ADHD in psychoeducation. However, if you do that, it is very important that you're not talking about genetics as being deterministic, because this is what we often hear, right? This is, my genes make me do it, I cannot do anything different. But as I showed you, this is not the case for ADHD. Genes are important, but they are not all. They are influenced by environmental factors, and there's possibilities to work with your heritable burden for ADHD to improve based on environmental settings. Biological insights from genetics may also help to define new treatment options. And here I showed you the data that from the biological studies, and you see that, for example, synapse function and the dopaminergic circuits are here, which we already treat in the existing medication for ADHD. But there's also GABAergic neurons, glia, and circadian rhythm that may be new targets for other types of medication or other types of treatment. Genetics may guide the individualization of treatment strategies. When should we treat? How should we treat? What we need for that is that we can translate the findings from big data approaches to measures that are meaningful for an individual, so biological markers that are meaningful for an individual. And what has been developed here is a polygenic score, a polygenic risk score, where we sum up the burden of an individual for ADHD, for example. So from this Manhattan plot, from this GWAS finding, we get one score per person, which reflects the risk for ADHD. However, at the moment, this is not sufficiently useful, sufficiently informative to use in diagnostics. We have to do larger studies, and we're busy doing those. And genetics may help us prevent impairment in ADHD. If we can predict, for example, who is particularly vulnerable to comorbidities, we may be able to prevent the comorbidities by altering the lifestyle. For example, cardiovascular and metabolic diseases. I said before that ADHD is not only biological. It is a construct that has a bio, psycho, social, and societal component. So here we asked, Ingji Shi, a PhD student in my group, asked the question, what is the effect sizes of different factors that contribute to ADHD? And in this case, she didn't look at ADHD, but she looked at externalizing problems. And she used one of the resources that is freely available on the web, the Adult Brain Cognitive Development Study, the ABCD study. She defined externalizing problems and internalizing problems based on the CBCL, and then looked at different risk and protective factors. So for example, polygenic scores for different psychiatric disorders and for different traits. She also looked at family history, so not only that has genetics, but also other types of family environments, for example, feed into that. She also used measures on lifestyle, for example, from objective measures like the Fitbit that is available there, and also from subjective report of the parents, And she looked at environmental factors at different levels, for example, the family, the school environment, and also neighborhood. And this is what she finds. So this is a very complex slide where she put together all the different risk factors and protective factors. So what you see here, for example, are the polygenic risk scores that have a relatively small effect size still, smaller than the family history that somebody has. So there's much more than we can measure at the moment with polygenic scores in the family. The Fitbit measures gave us a little bit of information, but if you, and especially sleep seemed to be important, but if you look at sleep reports from parents, this has a much larger effect size still. And then family and environment also play an important role in terms of increasing the risk, but also in decreasing the risk and being protective. So what you see here is essentially all the different layers that play important roles when it comes to risk for psychiatric diseases. And what you see is that not only biological factors, but also many environmental factors protect you or increase your risk for a disorder like ADHD in this case. So coming to an end with take-home messages. For me, as a neurobiologist, these are really, really exciting times. There's so much data sharing now out there. There's so many tools, so many methods, so much infrastructure out there that people are now willing to share, that we can make great strides in understanding faster what ADHD is biologically. ADHD has correlates in the brain, but there's a lot of heterogeneity there, and the small effect sizes do not allow us to use that as a diagnostic marker. ADHD is highly heritable. The genetic component is complex, and there is interplay between genetic factors and environmental factors that can work in both ways to exacerbate the problems or to ameliorate problems. Insights into the genetics and biology of ADHD can support psychiatric healthcare in different ways in the future, and things could be treatment, innovation, precision diagnostics, and prevention of impairment. What I would like to say for future research is that we are focusing very much on case control designs, on symptom counts, but what is much more important is that we also help understand everyday functioning of people with ADHD. And then, as I said, we should not only look at genetics or biology, we should integrate that with the other factors. And there should be a modernized concept of ADHD that could look something like that, where we are not only looking at risk factors, but also at protective factors and resilience, where we substitute only looking at symptoms by adding functioning and well-being to our analyses, look at strengths and weaknesses, look at biological factors, as well as behavioral descriptions. Not binarizing ADHD, but look more dimensionally and help or have the patients be involved in decision-making. And using digital interventions, as we just heard in the keynote as well. So with that, I would like to thank the people who are involved in this work. Many of the large international consortia that we work with are important for this work, and also people from my own group, as well as from other departments in Radboud UMC. Thank you very much for your attention.